Hi, everyone. Welcome to The Issue on the ASGCT Podcast Network. I'm your host today, a little bit of foreshadowing, Emily Walsh Martin. And today is a very special episode as I'm joined by two of my former colleagues, Jason West and Sanjay D'Souza, to do a deep dive into the ASGCT annual meeting abstract book, all nearly 5,000 pages of it. Now, I'm going to let them each introduce themselves in a second here, but the key information to give our listeners is that Jason, Sanjay, and I go way back. In fact, it's a decade to when we all worked together at CRISPR Therapeutics.
Yeah, Emily, thank you for having us.
Thank you for
No,
having
it's
us, Emily.
great to have you guys. And it's funny thinking back to those days at CRISPR, like the field has evolved now from when we all worked together.
Absolutely. It was such a delight to go through the abstracts with you for this conversation and think critically about how far the field has come since our days at CRISPR together. And really excited for the meeting here in Boston in mid-May. On our home turf, we are all Boston-based when Emily isn't sailing across the ocean. And so really excited to highlight to the listeners what we're excited about for the meeting. Obviously, we can't cover all 5,000 pages, but we'll do a short highlight reel of what we're excited about.
Exactly, kind of an amuse-bouche, if you will, for what's to come at the meeting. But before we dive into that, Sanjay, why don't you introduce yourself to the listeners?
Yeah, thank you, Emily. It's so much fun to reunite with you and Jason. We met and worked together at a time when CRISPR was so new that making a durable drug out of it was a bit more than a pipe dream. So I'm really excited to work with you guys again, and also excited to work with the ASGCT, who have impacted such great innovation in the gene and cell therapy space for so many years. So my background is in DNA repair, and that's when I joined CRISPR Therapeutics so that I could apply my training to this brand new technology a decade ago. And CRISPR being a technology that relied on the repair of DNA after engineered nicks or cuts, I felt it was a terrific opportunity to be part of that kind of innovation. So I started in the early discovery and platform group at CRISPR Therapeutics with Jason, where I worked on making the technology better. For example, improving the frequencies of homologous recombination, and then transitioned to the hematology group with Vertex as our collaborators. I then moved to the in vivo group at CRISPR Therapeutics, where we targeted the liver to edit genes using lipid nanoparticles as delivery vehicles. And finally, I was part of a program team that led the development of cardiovascular drugs. And my primary responsibility was to lead the preclinical development effort from guide discovery and off-target assessment into the non-clinical pharmacology and toxicology package. And as the programs matured, I also partnered with the CMC and regulatory teams. And this is an in vivo editing technology targeting the liver. And I'm excited to say that these cardiovascular drugs are now in phase one and phase two clinical trials. So I'm really excited to be part of this podcast with you and Jason, and looking forward to many more productive and fun interactions with both of you.
Absolutely. And Jason, why don't you share your background and introduce yourself?
Oh my goodness, I'm afraid that you're not gonna let me be a part of this after Sanjay's impressive background and overview. Hopefully I make the cut. Hopefully I make the cut. Thank you, Emily. Yeah, so Jason West, and as I mentioned, I'm Boston-based. I grew up in West Virginia in a post-industrial steel town. Did my undergraduate studies at West Virginia University, and then moved to Boston for graduate school training, postdoc, and then entered into the biotech industry over a decade ago. And I've worked across many different modalities during that time, including biologics. I perform small molecule screens to try to increase the potency of cell-based therapies. I worked on cell therapies, of course, including contributing to the work on sickle cell disease at CRISPR Therapeutics. Before CRISPR, I worked at Biogen, collaborating with Sangamo Biosciences' zinc finger nuclease technology. Worked on TALENs as well prior to that at Amgen, trying to engineer embryonic stem cells and induce pluripotent stem cells to model disease. Since my time at CRISPR, I've been working primarily in the AAV gene therapy space, but in my current role as Vice President of Global Research at Cambridge Pharmaceuticals, we have a rich pipeline, several commercial drugs, and so my role has oversight over enzyme-based therapies, biologics, AAV-based strategies, and everything in between. We've been trying to expand our pipeline, so it's been really exciting in my role, to see so many different technologies and think critically about, how those technologies could be developed for hopefully safe and efficacious treatments of rare diseases or potentially broader diseases. So, thank you, Emily, for inviting
Yeah,
Sanjay and I. Looking forward to diving into these abstracts.
Yeah, and
absolutely.
I'd like to point out that Jason's pioneering work at CRISPR in the hematology work enabled us to use expertise in DNA repair to get levels of homologous recombination that the field really hadn't seen before. It was such an exciting time then, and it was really due to Jason and his team's work that we were able to get to those levels of editing at that time. So it's really exciting to reunite and talk about those days.
Yeah,
And
absolutely.
as we move forward. So
Yeah, and I always joke, Sanjay and I co-led a department at CRISPR Therapeutics, and I used to tell my wife that Sanjay was my work partner. And now Emily has brought Sanjay and I back together here. So I'm
Exactly.
excited to
Reunited.
yeah,
I,
reunited. Excited to partner with you again, Sanjay.
Indeed, indeed. And I think it's important, to share with our listeners that I do, there's a bit of dramatic foreshadowing in having this episode, and in part because this episode is momentous. After many years that I've been working closely with the ASGCT, and since, 2.5 years ago when I incepted and originated the issue for the ASGCT podcast network, I'm happy to say that I'm handing over the batons as the host to Jason and Sanjay at the end of the podcast today. I'm thrilled to pass the baton because I think you guys are going to have unique and interesting insights to share with the audience as this podcast moves forward. I'm also excited, as Jason sort of mentioned, to do a little more sailing. So this all works out for everyone, and I'm leaving the audience in very good hands. Before I sign off, though, from the network airwaves, the key thing is we need to walk through the 2026 annual meeting abstracts. Now, we've each gone through the 4,000 abstracts in some level of diligence. I'm not going to say we read them all. And it would be impossible to cover them all in such a short podcast, but we're gonna do what we can, and we each sort of approached the abstract book with some themes that we were thinking about. And Jason, I think you've offered to go first and share your abstract theme of big genes. So why don't you go ahead and take us through?
All right, Emily, thank you. And yeah, thanks again for having us on the issue. It's been amazing listening to this over the years. And I think it's really having impact to the field as you dive deep into issues for the cell and gene therapy field. And I look forward to continuing those conversations with Sanjay and hopefully having you back as a guest of honor. Looking forward to the ASGCT meeting. And as you said, that on the theme of big genes, in my current role, I am working on muscular dystrophies caused by mutations in large genes that wouldn't fit into a single AAV vector. And perhaps because of that, current focus in my work, when I read through the abstract book, I was really excited to see some clinical updates on programs that are addressing high unmet need patient populations whose diseases are caused by mutations in large genes. And so the first abstract I'd like to highlight is abstract number 1. I didn't just choose that because I'm lazy and didn't want to read through the 5,000 pages. It's part of the presidential symposium, and I think it's going to be a really high impact presentation that'll be elicit a lot of conversation, and we'll dive into some of the details for why that is in a second. This abstract is from Encoded Therapeutics and their clinical colleagues who have been bringing forward their ETX 101 program for Dravet syndrome, which is a devastating neurodevelopmental disorder that includes, severe epilepsy. And this abstract will present the interim results from the Polaris phase 1/2 clinical trial. And what this is providing is an update on ETX 101. And they've treated 21 patients thus far. Before I get into some of those details, I first want, the big gene part. So Dravet syndrome, as I mentioned, is a devastating disorder, and it's caused by mutations in the SCN1A gene, whose protein coding sequence is encoded by over 6,000 nucleotide a stretch of RNA. And so that is something that can't be packaged efficiently into a single vector and delivered to the CNS where this defect needs to be repaired. So Encoded is not actually attempting to do that, encode the large transgene in a single vector. They're using AAV9 to deliver a transcription factor which can upregulate the SCN1A specifically in inhibitory neurons, which is important for the efficacy and safety of the therapy. And because about 90% of Dravet patients are their mutations cause haploinsufficiency, there is this locus that could be amenable to upregulation. And so, so much decades of work have gone into understanding this complicated biology and the transcription factor that can be delivered. And it's exciting to see that investment in the basic science, the translational science, the AAV gene therapies come to fruition here in the ETX-101 program. So their update will include, the clinical progress, which includes treatment of up to 21 patients, I believe, at the time of the abstract being submitted. And, thus far, I would say the results look pretty encouraging. So definitely looking forward to that abstract.
Absolutely. No, I think that's a great abstract. And it's interesting, right? Like sometimes you can't go through a problem. Sometimes you have to go around, and it's very, the approach they're taking seems very elegant, and very much looking forward to that data.
And I know epilepsy is something that you've really championed, Emily. You sailed across the Atlantic Ocean to raise awareness for epilepsy and blogged about it on the ASGCT website, something that people can still find. Now, I want to also highlight that this abstract is noteworthy not only for the clinical progress, but I think there's going to be a lot of conversations around one piece of the data presented that isn't directly related to the AAV9 gene therapy itself, but rather the immune conditioning often used to enhance the safety of administration of AAV gene therapies. And during this clinical journey, the investigators have applied both prednisolone alone or also prednisolone or sirolimus in combination. And one real big surprise in their abstract is that they have data in 6 patients who received that combo of prednisolone and sirolimus also known as rapamycin, suggesting that has impaired or dampened the function of their drug in patients. They speculate that this may be because rapamycin suppresses protein translation, and that may somehow be affecting how well their drug is working, but obviously really provocative data and so fundamental to what so many groups are doing with AAV gene therapies that it'll be really important for the field to understand what the data is and isn't and the ramifications for many clinical programs.
Absolutely.
Okay. Also on the subject of Dravet, Stoke Therapeutics and their clinical colleagues in abstract number 142 has an ASO technology that can upregulate the protein. They're presenting an encore publication of the clinical data applying their drug to Dravet patients. So definitely an opportunity for questions, conversations around those data, which is something the whole field is looking to for interesting application of ASOs for upregulation of a protein through splicing mediated mechanisms. So really cool work out of Stoke applied in the clinic. And then quickly, while also on the subject of large gene diseases, Stargardt disease causes blindness in kids as well as adults, high unmet need caused by the ABCA4 gene, which is also too large to fit into single vector cassettes. Ascidian in abstract 115 will be presenting preliminary safety data from their first in human trial that uses AAV8 to deliver an RNA editing cassette that repairs the first 20 or so exons of the gene at the RNA level. So a really interesting technology advancing to the clinic from Ascidian. And then also in the poster section, Huida gene will be highlighting clinical data from their investigator-initiated trial in Stargardt disease in China, where they've treated 6 patients with a dual AAV vector approach using an INTENE technology to deliver the full ABCA4 gene. And obviously, this is a sort of a new horizon for AAV gene therapies, these dual therapies. There's multiple academic groups and now biotech companies pursuing these technologies. So it'll be really exciting to see what Huida gene has been able to achieve in their clinical testing of their AAV-based INTENE program. I would also like to acknowledge the abstract from the EFPIA Working Group, abstract number 133, Non-Clinical Toxicity Study Duration in AAV Gene Therapy Development. Evidence from Industry Survey supports adequacy of short-term assessments. They also published this work in Molecular Therapy Advances back in December, in ASGCT journal, and they're doing a follow-up presentation at this meeting. And I think this is really important work studying the non-clinical data and now clinical data for multiple gene therapy programs to understand what is needed to articulate the potential risk and benefit of an AAV gene therapy in the preclinical space. And in their paper and in this abstract, the authors argue that potentially shorter toxicity studies will likely reveal everything that the model can reveal about the safety of a therapy, and obviously represents a potential method for companies, strong data support for companies to speak to regulators about what is necessary for their programs, and could represent an opportunity for companies to accelerate their preclinical development timeline, which is something so important for the way that programs are funded. So obviously, a potential high impact abstract from this group that will be read and discussed by many colleagues in the field.
Absolutely. Yeah. And I like how you phrase that, to the degree that a model can tell you about tolerability or toxicity concerns, that shorter term studies may be just as informative as longer term. It's important data, and definitely looking forward to the discussion that occurs on that. All right, Sanjay, tell us about your theme. Take us through what you've got.
Thank you. Thank you, Emily. We are in the next wave of innovation, I feel, and my theme was in vivo gene editing. And just to take you back a bit, in the early days of CRISPR-Cas9, the field was focused on proof of concept. Right? Number one, could we precisely edit human DNA? And number two, could we do so in a therapeutically meaningful way? So that vision crystallized through the pioneering work by CRISPR Therapeutics and Vertex Pharmaceuticals, who advanced ex vivo editing of hematopoietic stem cells for hemoglobinopathies. That work culminated in Casgevy, the first approved CRISPR-based therapy for sickle cell and beta thalassemia in December of 2023. And this was a watershed moment. Not just therapy, but proof that genome editing can deliver durable curative outcomes. But let's be honest, the ex vivo approach, while powerful, remains complex. It requires cell collection, conditioning, and specialized infrastructure, all limiting scalability. So the next frontier in genetic engineering is converging, bringing in vivo genetic medicines that you can administer directly into patients, and to not just hematopoietic stem cells, but also to cells of the immune system like T cells to fight cancer as well as autoimmune diseases. So it promises to be a really exciting ASGCT meeting this year, and I'd like to highlight a couple of developments in this whole theme of in vivo gene editing. Tessera Therapeutics have developed an RNA gene editor to correct the mutation that causes sickle cell disease using hematopoietic stem cell targeted lipid nanoparticles. They find that a single IV dose of these LNPs achieved an average of approximately 32% to 51% edited cells in non-human primates. They highlight their work in abstract number 13. At the same time, Intellia, who's also been a big player in vivo gene editing, are editing hematopoietic stem cells using liver-tropic LNPs by taking advantage of a developmental time window when the HSCs reside in the liver. And they were able to achieve robust HSC editing in neonatal as well as postnatal mice using their reporter constructs. So these are two very exciting in vivo gene editing technologies that are being developed by these two companies. And in parallel, the CAR-T field is moving towards in vivo CAR-T generation, essentially turning the patient's own T cells into CAR-T cells inside their body. Sana Biotechnology, in abstract number 20, report a potent, safe, and cell-specific in vivo generation of CAR T cells in NHPs with something called SG293, a novel paramyxovirus fusogen engineered to target the CD8 receptor. It also has a CD3 coactivator protein on the vector, which promotes CAR expression and activity in resting T cells, and a novel CAR transgene design that attenuates CAR protein expression during vector production. Similarly, Dana-Farber, in abstract number 24, have designed and optimized a T cell target alpha retroviral vector for in vivo CAR T cell production. They've tested this in an in vivo mouse model and of metastatic neuroblastoma. And what they find was that in contrast to PBMC only treated mice, all mice that received this novel vector showed expansion of CAR T cells over time and a pronounced reduction in tumor burden. And they also found no adverse effects, and the treatment was well tolerated by all mice. So many of these teams are now using these innovative models of delivery, like virus-like particles, LNPs, and alpha retroviral vectors to enable efficient in vivo gene editing. So I think that's the next wave of innovation as far as in vivo editing using these CRISPR components go. And it will be exciting to see how these the programs advance. And testing out these various delivery modalities will be key to determining how safe these drugs are going to be and how effective they're going to be.
Absolutely. So exciting to see so many abstracts in this space. And like you, Sanjay, I was really excited to see the primate data that Tessera, I'm not sure how to pronounce it, but the NHP data that they presented for in vivo editing of HSCs is really exciting. So really looking forward to seeing some of the details in that abstract. And I'm sure, again, it'll be a very high impact presentation. And there's so many, I think you couldn't highlight them all, obviously, because there's so much work going on in this area. It's really exciting to see those advances.
Absolutely. And I totally agree. I think one of the things that, the field has learned is that all of those steps in the process that Sanjay sort of elaborated, as you began to talk, of the cell collection, the conditioning, et cetera, those are absolutely integral to making the current therapies work, but are such a block to access for many patients because sometimes it takes multiple collections, et cetera, to get yourself to a final batch of cells that again could provide quite striking benefit to the patient. And so seeing these advances in vivo work is just amazing to see, and it'll be important to watch moving forward.
Reflecting back, we were talking at the beginning of the conversation about our careers, and I remember being at Biogen back in 2015, working on ex vivo sickle cell programs, and my amazing boss Kai-Shin Cheng there proposing in vivo editing as a research program to undertake, and us not really finding the footing or the mandate to prioritize that effort. And so it's been something that I've been wanting to, try to contribute to since that idea was planted in my mind, so many years ago. And it's exciting to see all these experts advancing these ideas towards in vivo editing, which can have such a dramatic impact on patients. So really excited to see the field continue to evolve their approaches for methods that can benefit patients.
And it's really exciting to see, from all these highlights that we presented, right? There's no one modality fits all approach, right? There are large genes, there are ASOs, there are different kinds of vectors, all of these trying to solve a particular problem, right? In gene editing and gene therapy. So it's really exciting to see all of these converge, right, at the same time. And ultimately, it's patient cures and patient efficacy and safety that is going to be the biggest barrier. And I think using this multimodality approach with these various levels of innovation and entrepreneurship is what we're going to determine how effective these drugs are going to be. So it's really exciting to see so many different technologies pioneered in this ASGCT meeting.
Absolutely. And I think, in fact, Sanjay, this segs nicely into my section. because I think it's only at the ASGCT annual meeting that you can bring together all these therapeutic modalities and have someone who may be super deeply focused on, for instance, lentivirus get exposed to AAVs or virus-like particles or alpha retroviral vectors, and start to see the possibilities where perhaps, they might be seeing some limitations in their current programs. And so what I think that means to me is that, now even more than ever, it's really important for us as a field to use this meeting to its full potential. And so my two themes that I'm going to talk through today is number one, hidden gems. That's a quick little vignette, but it's something that has annoyed me for a long time, and now we have some answers, some great work coming out of the Children's Hospital of Philadelphia there that I'll talk about. And then the other part that I guess we could call it long-term data, we could call it Emily's typical soapbox, but we'll dive into three abstracts that I found that I think are sort of crucial for us to understand not just the initial promise, but rather where the rubber meets the road, some longer term clinical data and outcomes that I think are important for us to keep focus on. So first, let's jump into the short vignette, and that is abstract 2384, which is an analysis of a small cohort of caregivers following AAV systemic administration and looking at whether or not they seroconvert. Now, it is a small cohort. I think it was 12 caregivers of patients who'd been given Zolgensma and 9 who'd been given Elvidus. But what's nice about that is it actually was pretty intensive caregiving. So for many of these caregivers, the patients were quite young. And so there was, for instance, assistance with toileting or diaper changes and that sort of thing. And so they looked to see if there was any evidence in those caregivers of seroconversion or of increase of anti-AAV antibodies. And to give away the answer is no, they did not. But I think this is a section of the IND that sometimes teams struggle to fill out in terms of transmission, shedding risk. And I think this is such an important abstract from Sean Carrig of CHOP that folks should pay attention to.
Really interesting one, Emily, and maybe not directly related, but another abstract that stuck out to me as well was from Ultragenyx abstract number 76, where they're presenting data about administration of AAV gene therapies into both preclinical models and patients with pre-existing anti-AAV8 antibodies. And that's another area where I think a lot of programs struggle because obviously when you begin your clinical testing, you want to set up your gene therapy for so much success, but excluding all those patients with pre-existing antibodies because of potential, safety, which is often debated. I think it's primarily, my understanding is primarily an efficacy issue of pre-existing antibodies affecting what your therapy can do. So I love to see that Ultragenyx is sharing that data with the field at the meeting. And like this abstract from the CHOP, as you mentioned, it's something that many programs deal with in terms of how do they include or exclude patients for their therapies. So an area of potential high impact.
And just to double click on what you said, certainly this is like the question of being able to administer to patients who are seroconverted for your serotype of AAV has been around since it seems the dawn of time, right? And certainly we've seen some groups present like around AAV5, which seems to be a little weird and interesting and maybe not susceptible to that phenomenon. Also about adenoviruses, which also may suggest whatever it is, whatever magic it is. Maybe it's the kinetics of transduction, who knows? But we've seen that, but it's great to see Ultragenyx also bring their data forward. These are the kinds of, I think, abstracts that really do help the field as a whole move forward.
And also, it's cool that it's also highlights the caregivers, right? And
Mhm.
caregivers are very often the overlooked part of a lot of these treatments because they are in very close contact with the patients and are the primary means by which the patients interact and see them every day. So it's really nice to see them also highlighted and try to figure out what happens to them as part of this course of treatment. So.
Absolutely.
Yeah, and and also I've been a part of conferences, and I'm sure this isn't just specific to the DMD field, but conferences where parents are talking about having two affected children and wanting to try to get both children in a clinical trial at the same time because they don't want to risk the sibling developing antibodies that would prevent them from receiving treatment later down the
Mm.
road if the first sibling receives therapy. So this can have also broader impact on families that, have multiple individuals that might be
Yeah,
eligible for gene therapies.
absolutely. Absolutely. It's a great point. Okay, and so then my last sort of tranche of abstracts are all sort of longer term follow-up studies. The first one that I want to highlight is from the Telethon Institute. It's abstract 145, looking at lentiviral gene therapy and beta thalassemia. And here, I think what's interesting is that they're looking at that sort of long-term follow-up, I think up to 6 years in some cases, and also variability of efficacy. And so they have some interesting new hypotheses for what might determine efficacy with this approach of therapy. But, they're reporting 6 patients, 2 of which failed to achieve sort of transfusion independence at all. 2 others that lost that benefit after 5 to 6 years. And so important data for us to understand because I think when we think about running these trials, the hope is that we can give every patient the best shot on goal to have a benefit. And so understanding where things maybe didn't rise to the level of efficacy we were hoping for is super important.
Absolutely. Anytime physician scientists or the scientists from the Telethon Institute present, I try to attend because it's
Right.
the collective wisdom that's applied to their programs and the impact those programs can have is so high that I always want to be in the audience for those presentations.
Yeah, and
Yeah.
the lead author there is Maria Rosa Lidanici, I should have said.
Yeah, and these HSC biomarkers have long been the holy grail of HSC biology and
Yeah.
engraftment and curative efforts for sickle cell and beta thalassemia. So it's really exciting to see this work sort of presented. And as Jason said, yes, I always also attend their talks because they've been at it for a really long time, and it's getting better and better. Our knowledge of biomarkers and the therapy is getting a lot better over the years. So,
Absolutely. Okay, next up, I have an abstract from Sanofi on their now terminated program, SAR 444836. It's abstract 1357. The lead author is Fatih Ezgu. And so this is the phase 1/2 study that they're publishing. It was a liver target AAV looking to treat PKU. They studied 2 dose levels, generally saw safety and tolerability, but did not see the level of clinical benefit on phenylalanine levels that they were hoping for. And so even tripling the dose didn't improve the efficacy in 2 patients who received that dose level. And so they've terminated the study. But I think, again, not to belabor this, but this is the kind of data that needs to come to ASGCT, whether it's good or bad. Understanding the long-term outcomes after treatment and sharing that data helps to move the field forward. And maybe I'm I don't think I'm alone in having this opinion, but right now we don't necessarily know how to redose patients after they've received an AAV therapy. And so when a patient comes to us and they consent to be part of our trial, there's a social contract that we enter into as the sponsor, and in part, like when they become part of our trial, they realize it's sort of their one shot on goal. They're willing to take that sort of chance with us. And I think their expectation is that maybe they'll get some benefit, but more importantly, that their data, the information from treating them will benefit the field. And so it is our obligation, in my opinion, as sponsors of these trials, to make sure that their data gets represented and circulated and shared so that the field can understand better, contextualize, and hopefully do even better in the future if needed. So again, that study's from Sanofi, and kudos to them for bringing that here. And then my last abstract is, again, similar theme. It's abstract 3378. It's a comparison. It's a very small study. So two siblings with GM1 gangliosidosis. This is from Hera Akmal at NHGRI, and it was an AAV therapy. And they sort of talk through the outcomes for these two siblings. And I think what was useful about this is it does show the complexities of study design for diseases where there is sort of variable progression, variable outcomes, trying to understand the value of how do you get the right natural history data set to do a proper comparison and assess things. So again, kudos to them for bringing this data forward.
All right, Emily, thank you for those. I'll definitely be making an effort to attend those. Really excited to see all of that clinical progress. Of course, one thing I wanted to say is, I think out of those 5,000 pages, so much work that we couldn't acknowledge in the time of this podcast. And obviously there's a little bit of a bias towards these clinical stage programs because we're seeing the hopeful payoff. Obviously sometimes we take a few steps forward, a few steps back, but obviously we're all here because we want to impact, health and disease in a positive way. Would also like to, shout out to all of the abstracts that we aren't acknowledging that are contributing to the field, whether it's, fundamental discoveries coming out of academic research labs, CMC or manufacturing progress to make higher quality or lower cost of goods. Maybe I'll just do a quick shout out again to Ultragenics, and they have an abstract 129 using CHO cells to try to manufacture AAVs, which I don't know anything about it, but it might, may work, may not work. But obviously we all want to see the cost of goods come down so that these therapies could get into the hands of as many patients as possible, and also create a sustainable business model for the investors and companies that are spending so many dollars bringing these therapies forward. But obviously, so much work and really excited for the meeting. Sanjay, any last thoughts from you?
Yeah, thanks, Emily, for highlighting the patients, especially because, as Jason said, I think it's the patients are these therapies are most meaningful to the patients, right? And getting to them as quickly as possible and economically feasible manner is the primary goal of the work that we do. And even if it's a two-sibling trial, right? If it's a small patient trial, it's still impactful and still meaningful because the learnings that we get from that are immensely beneficial for future therapies. So thank you for highlighting all of that. I think it's going to be a really exciting ASGCT, and I will make sure that I attend as many talks and read as many abstracts and posters when I attend it next month.
Absolutely. Take good notes for me. All right, so that brings us to the end of this episode. Oftentimes on the issue, we close with me granting the guests a magic wand. Obviously, you guys get to move forward granting magic wands as you like to your guests. I'm just curious, from reading through the abstract book, anything where you would love to take a magic wand and wave it to impact gene and cell therapy?
My magic wand would definitely be the manufacturing and scalability of gene therapies. I think if I could wave a magic wand and enable these manufacturing capability and scalability to be easier and cheaper, so that they could get to patients almost immediately. I would love for that to happen. I think there have been great innovations in the field, and I do think these things are actually possible. I can't wait for it to happen, and I just want it to happen like tomorrow, right? So that would be my
I love
magic
it.
wand for the future of cell and gene therapy.
Yeah, great question, Emily. I wish I had prepared for that question. I should know better to have not prepared.
Exactly.
But I can't help but think, because we're reflecting back, I can't help but look forward and think about, 2036 ASGCT and all of the progress that'll be made based on the fundamental foundational work being presented today and at past ASGCT meetings. Hopefully in 10 years, the word cure is able to be used with a straight face, or maybe I should put that more eloquently, but, hopefully in 10 years, the word cure is brought back in the gene therapy field due to innovation. I know currently, these one-time treatments, we hope that they're durable, safe, and efficacious. But we really need time to establish that as a field, obviously, right? So
Yeah.
looking outward, continued progress and presentations at ASGCT will define those risk benefit profiles, durability, and whether these one-time treatments can really change the lives of individuals throughout their lifetime. So looking forward to future years and looking forward to this meeting and seeing colleagues from the past and making new connections.
And I guess with my magic wand, I'm going to take a note from you and go back to that word durability. I think this is the cornerstone of the promise we're hoping to provide patients in this field is durability. And so the only way to know if you have something durable is to follow it for a long time. And the only way for the field to understand the implications of our therapies and the durability of them is to present those durability data at the ASGCT annual meeting. So my magic wand is that, this year, but also next year, I would love to see, we should be able to get 150 abstracts of greater than 4 years, right? Of follow-up. That just should be easy. Let's
Yeah.
let's see that. Let's have 150 abstracts come in for 2027, all with 4 years or greater follow-up. I'd love that. That would make my day.
Yeah, that would truly be amazing,
Wonderful.
really amazing.
Yeah.
All right. Well, thank you both, and very much good luck to you guys. I know I'm going to be listening in as you guys have your podcast conversations, and really, really looking forward to the insights that you and your guests bring to our field.
Thank you, Emily.
Yeah, thank you, Emily, for passing the baton. Hopefully we don't break it.
You're going to be great. You're going to be great.